So this is the competency. Now gastrointestinal physiology has about 10 or 11 competencies in your new curriculum. So today I will be starting off with your competency number 1, that is 4.1. Describe the structure and functions of digestive system. Right? So I would be dealing with this today. Now this is a simple topic, but it is important as far as your exams are concerned. And secondly, as far as your practice as a physician is also concerned, right? Because there would be a lot of gastrointestinal disorders which you will come across in your day-to-day practice as a doctor. So in the describe the structure and functions of digestive system, basically I would be dealing with the structure of the GIT, the functions of the digestive system, and I will go into a little bit of detail about it. In the structure, I would be telling you about the cross-sectional structure, the various layers, what is the blood supply, what is the innervation of the digestive system, functions, again, what are the major physiological processes which are going on in the digestive system, right? So GIT, by and large, it comprises of the gastrointestinal tract along with the associated glandular organs. Just a second, please, okay? So gastrointestinal tract, it is a series of hollow organs which stretches from your oral cavity from mouth to the anus about 15 feet long. There are glandular structures or organs which are not actually a part of the gastrointestinal tract, but they are secreting their secretions into the GI system by means of the connecting ducts. So together they form the GI system, right? So here you can see that this is showing you the gastrointestinal tract along with its associated glands. It starts from the mouth. From here, it continues from the mouth. It continues as the food pipe or the esophagus. Within the mouth or the oral cavity, there are presence of salivary glands. Esophagus, it continues or ends into the stomach, which leads into the small intestine, the large intestine, and there are various parts of it which you are already familiar with. In the associated glands, you have primarily the parotid gland, which is a type of salivary gland. Of course, you have submandibular and sublingual, and there are accessory salivary glands as well. Another associated gland is the pancreas, which by means of the pancreatic duct would be, you could say, secreting into the duodenum. Okay, its secretions would be reaching the small intestine. Third associated gland is the liver along with the gallbladder. By and large, the functions of the of this GI system, you can enumerate them as digestive and non-digestive functions of the gastrointestinal system. So functions of the GIT, the main function, of course, all of you are familiar with, is that digestion and absorption of nutrients and water. Right? So the overall function of GIT is to absorb nutrients and water into the circulation. Nutrients, once they have been digested, they would be absorbed, and the absorption will be accompanied by absorption of water. Second function of GIT is to eliminate the waste products Now, the water-soluble waste products are primarily excreted from our body by means of the renal system. So here it would be that it would be excreting out primarily those substances which are not water-soluble, which cannot be excreted out through the kidney or through the renal system. So it excretes or eliminates waste products which are liberated from the liver, such as the cholesterol, steroids, drug metabolites, and the fat-soluble excretory products. Another function of GIT is that it protects itself from the infectious microorganisms which can enter along with the food and water, right? So because GI system, it is connected to the exterior or to the external environment. We ingest food by means of the mouth and it reaches the oral cavity, which is having, you could say that it is in direct contact with the external environment. And similarly, the anal opening is in direct contact with the environment, right? Or whatever we are eating, along with that, we can also ingest a number of microorganisms with it. So the GIT needs to protect itself from these infectious microorganisms which can enter along with the food and water. And how does it do it? The protective functions of GIT, of course, this I've told you, it is a long tube. It is in contact with the body's external environment, vulnerable to infectious microorganisms. It protects itself by means of a complex system of defense, and this consists of immune cells and other non-specific defense mechanisms. In fact, the GIT represents the largest immune organ of the body. So here I have just enumerated a few examples of how the GIT plays a role in protection, or it plays a role in the immune system. So saliva, it contains mucin, it contains immunoglobulin A, it contains lysozyme. All these prevent the penetration of pathogens. Gastric juice, it is highly acidic, it has a bactericidal effect. In addition to this, our intestine, it has an intestinal flora within itself, which again is very important for the protective function of the GIT. It prevents the spread of pathogens. There is also presence of the lymph tissue, the immunocompetent lymph tissue, along with the Peyer's patches, which helps in the immune function of the GIT. So this is just an introductory aspect of the GIT. Now coming on to the physiological processes or the digestive functions of the GIT, right? So I told you in the previous slide, I told you that the functions of the gastrointestinal system can be digestive and it can be non-digestive. In the non-digestive functions, you have the protective function of the gastrointestinal tract. Now, when we talk about the digestive functions of the gastrointestinal tract, then there are certain physiological processes which are going on within the tract, which helps in the digestive function of GI system, right? Till now you have already had lectures on almost all the systems of the of your body, right? So you are familiar with quite a number of terms. So when we talk about digestion, here we are talking about what is digestion. Digestion is basically that whatever food you are ingesting, it is in the form of large macro molecules which cannot be absorbed into our bloodstream, right? So it has to be first broken down so that the digestive enzymes can act on it. Once the digestive enzymes have acted on it, it is ready for absorption, right? So the first major physiological process is the digestion. Second one is absorption. Absorption is basically when the substance enters from the digestive tract into the bloodstream. जैसे आपने renal system में पढ़ा होगा absorption और secretion. तो secretion क्या होता है? Secretion होता है जब bloodstream से renal tubule में आ रहा था. यहाँ पे क्या होगा? Secretion कब बोलोगे आप? या तो bloodstream से वो it is coming into the digestive tract, or it could be the secretion of various juices which can occur from the GI system itself, right? Absorption would be when the substance, the nutrient material is taken up from the GI system or the GIT into the bloodstream. For all these processes to occur, there has to be the movement of the food particle, the movement of the particles which have been acted upon by various digestive juices. It has to move in the anal direction from the mouth towards the anus. So for that movement, motility is required, which is brought about by the interplay of the muscles which are present within the gastrointestinal tract. Okay, so the major physiological processes, this is just so that whenever I'm using these terms, you don't get confused with it. Motility refers to movements which mix and circulate the GI contents and move them along the length of the tract. So normal movement is from the mouth towards the anus. In case of deranged physiology, there can be a movement which occurs in the opposite direction also, when you can term it as the person might be having vomiting when there is the expulsion of the food or expulsion of the contents of the gastrointestinal tract through the mouth, right? So it is achieved by the coordinated activity of the muscular layer. It can be either in the forward direction or in the backward direction. What is the importance of motility? It serves in the mixing of food with the GI secretions, reduces the particle size, and moves the food content, moves the contents of the tract forward. Secretion refers to processes by which associated glands and mucosal lining of GIT release these kinds of substances into the tract. So it is primarily the water, the electrolytes, the enzymes, the mucus, the humoral agents. They are released into the gastrointestinal tract by means of the mucosal lining of the gastrointestinal tract or by means of the associated digestive glands. Digestion, it refers to the process by which the food and large molecules are chemically broken down to smaller absorbable molecules. Absorption, I've already told you that it is entering the blood by means of the from the GIT or from the GI lumen. It is entering the bloodstream. Okay. So why do we actually need to process the food which we ingest? Why is it required? So for the simple reason why it is required. It is required because ingested food is in the form of large particles. No matter how much we chew the food, but still it is unable to cross the intestinal epithelium, right? So gut allows transfer of only specific molecules below a certain size, and so it needs to be ingested, it needs to be processed before it can reach the circulation. If the digestive system is not able to process the ingested food particles, it may lead to an immune response, because then it will start acting as an antigen and it can lead to a diseased condition. All these processes are interdependent. This is self-explanatory. Digestion नहीं होगा तो absorption नहीं होगा. Digestion के लिए movement is required. Digestion can only occur if there is secretion, right? So absorption occurs only when the food is digested. Digestion occurs if there is secretion. Secretion here means secretion of various digestive enzymes which are present in the various digestive juices. So it could be the salivary secretion, it could be the gastric juice secretion, it could be the pancreatic secretion, it could be secretion of bile, it could be secretion of intestinal juice, and similarly, the processes can occur only if the movement of gut brings it to the proper location. So we have specific enzymes which are released at specific places, and it is only if they are present there can the enzymes act, because all the enzymes need optimum pH, they need optimum conditions before they can act. So that is just an overview of basically what the major physiological processes are going on. Now we come on to the structure of GIT. अभी structure of GIT में आपको एक short note आ सकता है, या you can get draw well labeled. It can be a part of a long question where you can just draw a label diagram of the structure of GIT, right? So the structure of GIT, although it varies from region to region, but there are certain common features which are present in the overall organization. So by and large, GIT is arranged in the following layers from inside to outside. So the innermost layer is the mucosa. Outer to that you have the submucosa. Then you have the muscular layer. And finally, the last layer is the serosa or the adventitia. Right? So this is showing you the general organization of the layers of GIT. Okay. So as you can see, this is the GI lumen. Here you have 4 layers. So lumen is towards the hollow cavity which is there. So innermost layer you have the mucosa. Here you can see in the cross sectional that these are the layers which are representing the mucosa. Outer to the mucosa you have the submucosa. Outer to the submucosa, this is the submucosa. Outer to the submucosa you have the external muscular layer, which comprises of 2 layers, the longitudinal muscle layer and the circular muscle layer. Outermost layer is the serosa or the adventitia. So this is the layer which attaches the GIT through to the abdominal wall by means of the mesenteries. So mucosa, it is the innermost layer, consists of 3 layers. So the innermost layer in turn consists of 3 layers. These 3 layers are, the innermost layer is the epithelial lining of the GIT. This epithelial lining again varies. As you can see that in the small intestine, it will be the epithelial lining would be thrown into a number of folds which would be known as the villi and the microvilli. Outer to the epithelium is the connective tissue which is rich in glands, lymph nodules and capillaries. So this layer is known as lamina propria. Outer to that you have the muscularis mucosae, which is the mucosal folds and ridges occur because of this layer. So 3 layers in the mucosa: epithelium, lamina propria and muscularis mucosa. There is specialization, as I told you, in the epithelial layer. Small intestine, it is thrown into a number of finger-like projections which are called as villi. They help in increasing the surface area because most of the absorption of nutrients occurs in the small intestine. Then you have the indentations or infolds of the epithelial lining, which is known as crypts. So these are the crypts which are present in the small intestine. They are known as the crypts of Lieberkühn. The epithelial lining of the GIT is continually renewed and replaced by dividing cells. So this is, it keeps on replenishing itself in 3 days. About 3 days, it takes about 3 days to renew itself. These proliferating cells are located in the crypts. That is, the zone of intestinal stem cells is present in the crypts, right? So just if you compare the small intestine with the large intestine, that is colon, you can see the difference in the epithelial layer. You can see that in the small intestine, there are numerous finger-like projections which are called villus, whereas in case of the colon, there are the projections are not that much in number, because in the colon it's primarily the absorption of water which occurs. Nutrients are absorbed in the small intestine mainly. So this is just again a structure of the small intestine. It is showing you the presence of, this is showing you the villus, then this is showing the direction of cell migration and maturation. It is formed in the intestinal stem zone cell, which is present in the crypts from where the cells migrate and they are shed and renewed. Okay. These crypts of Libacon, they contain a number of specialized cells, which I will deal with when I'm talking about the intestinal juice secretion, right? So for now, you can just remember that there are 3 types, mainly 3: the goblet cells, the argentophane or the enterochromaffin cells, and the Paneth cells. The vascular system for absorption of fluid and dissolved material, it is going in the form of a branching system which is going into the villus, and there is a lymph vessel, the central lacteal, for absorption of fats into the lymph. Second layer is which is outer to the mucosa is the submucosa. So in the mucosa also you have presence of loose connective tissue which is called lamina propria. In the submucosa, it is a layer of loose connective tissue per se, where there are the presence of the major blood and the lymphatic vessels, as well as there is presence of the larger nerve trunks which are present, and there is also presence of a submucosal or the Meissner's nerve plexus, right? So here you can see that this is the outer layer which is there, which this is the innermost layer, which is the mucosa. Inner to the outer to the mucosa, you have the submucosa, which contains the blood vessels, it contains the lymphatic vessels, it contains the large nerve trunks, which would be the sympathetic and the parasympathetic, and then it also has the presence of the submucosal or the Meissner's plexus, which is a part of the enteric nervous system. Outer to the submucosa, you have the Muscularis External. So outer to the submucosa, you can see there is the layer of external muscular layer. Now this muscular layer in turn comprises of 2 layers of smooth muscles. You have the circular muscle layer and you have the outer longitudinal muscle layer. So innermost is the mucosa, outer to that you have the submucosa, outer to that is the Muscularis External. This is where the circular muscle layer and the longitudinal muscle layers are located. In between the 2 muscle layers, you have the presence of the myenteric or the Orbach's nerve plexus. So this again is a part of enteric nervous system. I'll just tell you about the enteric nervous system when I talk about the innervation, right? So this layer, it is responsible for mixing and circulating the luminal content. So when the longitudinal muscle layer contracts, it will lead to the shortening of the intestinal segments of the GI, and it will help in moving the food content or the contents of the GIT in the forward direction. Circular muscle, when it contracts, as the name suggests, circular, so it is going to cause the reduction in the diameter of the GI lumen, right? So it will help in the mixing of the contents, it will help in the churning of the contents which is present, breaking down of the contents. In addition to that, it has presence of nerve plexus, myenteric or Orbach's nerve plexus, which lies between the two layers of the smooth muscles of GIT. ठीक है, तो दो nerve plexus हो गए, एक हो गया आपका जो सबम्यूकोसा में प्रेजेंट है, सबम्यूकोजल लेयर में, सबम्यूकोसा और मस्कुलरिस एक्सटर्ना के बीच में, that is known as the submucosal or the Meissner's nerve plexus. Now, since the submucosa contains a number of glands, so this submucosal plexus would in turn be responsible for most of the secretory activity which is going on in the GIT. Second nerve plexus is the one which is present between the outer circular and the inner longitudinal muscle layer. So it would be responsible for mixing the contents. It would be responsible for movement of the contents or propulsion of the food along the GIT. Right? So this is just giving you the comparison between the longitudinal and the circular muscle layer. It is oriented in the longitudinal or long axis of the GIT. Its contraction which would cause the shortening and expansion of a particular segment. So it will help in movement of the food or propulsion of the contents of GIT. Circular muscle layer, the long axis is oriented in the circular direction. So thicker than the longitudinal muscle layer, it will exert a more powerful contraction, and its contraction will narrow the diameter of the lumen. So it will help in increasing the pressure also, and it will help in mixing the contents also. There are certain variations in the these Muscularis External. So just enumerated a few of them. At places, the circular muscle layer, it becomes thickened at places to form sphincters. Now, जो sphincter है, sphincters बहुत strategic locations पे present होते हैं gastrointestinal tract में. Now, these sphincters, what is the importance of these sphincters? These sphincters are important because they prevent the backflow of the contents. Now, you can just think for yourself that suppose the food is coming from the mouth, it flows into the, it moves into the esophagus. From the esophagus, it reaches the stomach. The stomach, you know, is having a highly acidic content within itself, right? अगर वहाँ पे कोई sphincter present नहीं होगा, तो क्या होगा कि stomach का जो भी content है वो it will keep on regurgitating into the esophagus, right? To prevent this backflow of the stomach content into the esophagus, we have a presence of the sphincter, which is known as the lower esophageal sphincter. This is just one example which I am giving. Okay? When I am talking about the motility, I'll go into the details of other sphincters also. So, ये sphincter क्या होते हैं? Sphincter कुछ नहीं है, circular muscle layer है जो thickened हो गई है at places to form sphincters. Stomach में, in addition to this, an oblique layer of muscle is also present deep to the circular muscle layer, which gives rise to the various folds within the stomach. In the large intestine, the longitudinal muscle layer, it is aggregated into three band-like structures, which is known as tinea coli. इसको ना worm से मत confuse करना, ठीक है? So this is showing you the large intestine. So in the large intestine, this is, you have the ascending colon, you have the transverse colon, the descending colon, sigmoid colon, rectum, and the anal canal, right? So here you can see that this is showing the longitudinal muscle layer. It is aggregated, or it is in the form of three band-like structures, and these band-like structures are nothing but longitudinal muscles of the large intestine, which are known as tinea coli. In between this, you can see there is bulging of the large intestine. These are known as the haustra or the haustrations. So GI smooth muscle, ये आपको जब nerve muscle physiology पढ़ाई गई होगी, तो you must have been told about the physiology of the smooth muscle, that there are two types of smooth muscles. You have the multi-unit smooth muscle and you have the single unit smooth muscle, right? So here in the GIT, you have the single unit smooth muscles which behave as a functional syncytium due to the presence of gap junctions. Smooth muscles, they make up all the contractile tissue of GIT except where you have the presence of skeletal muscle is the pharynx, upper 1/3 of esophagus and external anal sphincter, right? So you can get an MCQ which could be based on this. You could be asked in your viva voce examination also, that enumerate the areas in the GIT which has striated muscles, right? So here you have the three examples which are given, or you could have an MCQ also, which one of the following is not striated, or which one of the following is not, you could say, smooth muscle in the GIT. So in the electrical activity, this is just an overview. Again, you have the presence that in the smooth muscles, they do not have a resting membrane potential per se. Their resting membrane potential keeps on fluctuating, and these fluctuating resting membrane potential is basically the basic electrical rhythm which is there, which gives rise to the slow waves. When it reaches the threshold, then the spike potentials are produced, right? So this is just showing you the electrical activity of the GI smooth muscle, and this can be stimulated. That is, the number of spike potentials can increase if there is a stretch to the GI lumen or GIT, or there is the secretion of the acetylcholine or parasympathetic stimulation can lead to increase in the number of spike potentials. Sympathetic stimulation, presence of norepinephrine, they can lead to decrease in the number of spike potentials. Right? So this slow wave is initiated by interstitial cells of Kajal. Again, it is an MCQ question that the slow waves or the basic electrical rhythm in the GIT is initiated by what type of cells. So this could be one of the MCQ questions. Now coming on to the innervation of the gastrointestinal tract. So I think I am quite clear, and hope you all are not having any problems. Okay, so let's continue forward. Now coming on to another part of this structure and function of GIT, a very important part, which is the innervation of gastrointestinal tract. Now this can come as a short note itself, or you can get it as describe briefly the innervation of gastrointestinal tract, or you can just be asked about what is the nerve supply of gastrointestinal tract and give their functions, or what is their physiological functions. Okay. So it's innervated by two sets of nerves. One is known as the extrinsic nervous system. Another one is known as the intrinsic nervous system. तो nerve supply GIT की दो nerve types of nerve से होती है. एक जिसकी cell body GIT के बाहर होती है, which is located outside the gut wall. It is extrinsic to that gut wall, so it is known as extrinsic nervous system. Second is that it is present within the gut itself. It has the nerve cell bodies, the neurons, everything is located within the gut wall itself, within the GIT itself. So this is intrinsic to it. It is inherent to the GIT. It is known as the intrinsic nervous system or the enteric nervous system. It can function independently of the extrinsic nervous system. Agar extrinsic nervous system को काट भी दो, then also, even if there is a lesion of the extrinsic nervous system, then also the GIT can function to a certain extent because of the presence of enteric nervous system. Extrinsic nervous system, it helps to modulate the intrinsic nervous system functions. It helps to modify the activities or modify the regulation of the enteric nervous system. Right? So extrinsic innervation, what is extrinsic innervation? Extrinsic innervation is coming by means of the autonomic nervous system. You have, two divisions of autonomic nervous system. One is your parasympathetic division and another one is the sympathetic division, right? So parasympathetic innervation to the GIT, it is by means of craniosacral division. Craniosacral division. Craniosacral means cranial nerve है एक और pelvic nerves है. Cranial nerve में कौन सी cranial nerve है? It is by means of the vagus nerve, and pelvic nerve में you have the S2, 3 and 4 divisions of the sacral division. ठीक है? So this is your parasympathetic innervation. Sympathetic innervation, it comes from the thoracolumbar segments. Here you can see it is the thoracolumbar. In the thoracolumbar region of the spinal cord, the preganglionic neurons would be arising from the intermediolateral gray columns. So this you must have already read in your anatomy classes also, that it has the dorsal horn, the ventral horn, and the intermediolateral gray horn. So intermediolateral gray horn, thoracolumbar division, which is giving rise to the sympathetic supply to the GIT, and it will be reaching the prevertebral ganglia, which is your celiac, the superior mesenteric, and the inferior mesenteric ganglia, right? So these fibers, they project to the myenteric and the submucosal plexus of the enteric nervous system and help to modify the activity of the enteric nervous system. So parasympathetic is via vagus nerves. The cell bodies are present in the nucleus ambiguous and dorsal motor nucleus of the vagus nerve. Both these are present in the medulla. Efferent vagal fibers, they synapse with neurons of the enteric nervous system, and it supplies all these areas of the GIT. Proximal part of the lateral, proximal part of the large colon. Sympathetic innervation to the gut, it is located in the thoracolumbar region of spinal cord. I've already told you, starts from the intermediolateral gray columns. They make synaptic connections with neurons in the sympathetic ganglia. I've already shown you the superior mesenteric, celiac, and the inferior mesenteric ganglia, and they give projections to blood vessels, mucosa, and specialized regions of the musculature. Functions of parasympathetic and sympathetic system, what do they do? पैरासिम्पैथेटिक स्टिमुलेशन जीआईटी में याद रखो इंक्रीज करता है गट की और स्फिंक्टर की टोन को, राइट? वी आर टॉकिंग अबाउट द स्मूथ मसल. सो इन द स्मूथ मसल, stimulation of the parasympathetic nervous system will cause the increase in the tone of the smooth muscles of the gut, as well as it will increase the tone of the sphincter. This increases the frequency, the strength and velocity of smooth muscle contractions, as well as motor and secretory activities. Okay, so the function of parasympathetic nervous system in gastrointestinal tract is to increase the contraction, to increase the tone, to increase the secretion of juices. Sympathetic innervation क्या करेगा? Sympathetic innervation by and large इसका उल्टा करेगा. It would be opposite to what the parasympathetic innervation was doing. So it is going to cause a reduction in the tone of the muscles. It will cause reduction in the activity of the GIT. So the GIT, you can see, is sleeping here, reduces activity of the gastrointestinal tract. It inhibits the motor activity of the muscularis externa. What is muscularis externa? Muscularis externa is basically the longitudinal and the smooth muscle layer of the GIT. So it induces contraction of the muscularis mucosae. Muscularis mucosae is a layer of the mucosa. It causes reduction in the intestinal blood flow. तो अगर blood flow reduce कर रहा है तो ये secretion को भी कम कर सकता है, right? But main activity gut में जो होती है वो parasympathetic की होती है और enteric nervous system की. Sympathetic is there doing a role of a supporting actor. Okay. So here you have the innervation of the GIT. As I told you, innervation of GIT is by means of two sets of nerves. One is your extrinsic nervous system. In the extrinsic nervous system, it is coming by means of the autonomic nervous system. In the autonomic nervous system, you have both the divisions of autonomic nervous system, that is the parasympathetic as well as the sympathetic divisions of the autonomic nervous system, which is supplying the GIT. Second is the intrinsic or the enteric nervous system, right? So here in this diagram, you can see the presence of both the extrinsic as well as the intrinsic nervous system. So this is sympathetic and parasympathetic. Isme you have to remember that in the parasympathetic and sympathetic, when we talk about the motor divisions of the parasympathetic and sympathetic divisions, you are talking in terms of the preganglionic fibers, you talk about the ganglion, you talk about the postganglionic fibers, right? When we talk about the ganglia, we talk about whether the ganglia is located close to the effector organ or it is located away from the effector organ, right? So in the sympathetic division, as I showed you in the previous slides, in what was happening in that, it is starting from the preganglionic fibers are starting from the intermediolateral gray column, and then they make a synapse in the prevertebral ganglia, right? So you have the presence of three ganglia, that is the celiac, the superior mesenteric, and the inferior mesenteric ganglia, from where the postganglionic fibers will arise and they are going to innervate, right? Now, in this, what another important point which you should remember here is that the extrinsic nervous system is primarily ending on the neurons of, they are synapsing with the neurons of the intrinsic or the enteric nervous system. तो ये directly नहीं जाते हैं, ठीक है? GIT में what are the effector organ? Effector organ क्या होगा? Effector organ होगा आपका circular muscle layer होगी. Effector organ would be your longitudinal muscle layer as far as the movement of the GIT is concerned, right? Secondly, what would be an effector organ in the GIT? Another effector organ in the GIT would be your secretory gland, because they are going to cause the secretion of various substances, right? Depending upon what part of the GIT you're talking about, आपका वो secretory, वो effector organ, ठीक है? तो अब मेन एक्शन तो जीआईटी में इन्हीं का होना है कि मूवमेंट होना है और उसका डाइजेशन होना है. जब डाइजेशन हो जाएगा, इट हैज टू बी एब्सॉर्ब एंड व्हाटएवर इज रिमेनिंग हैज टू बी एक्सक्रीटेड आउट, राइट? तो ये कैसे होगा? ये होगा बिकॉज उसमें नर्व सप्लाई है. ये नर्व सप्लाई कहाँ से आ रही है? नर्व सप्लाई आपकी एंट्रिक नर्वस सिस्टम से भी आ रही है. एंट्रिक नर्वस सिस्टम को मॉडिफाई क्या कर रहा है? Autonomic nervous system modify कर रहा है, ठीक है? So here you can see that the sympathetic and the parasympathetic fibers, what they are doing is they are ending on, or they are synapsing on the myenteric plexus, which is present between the longitudinal and the circular smooth muscle layer, and you have the submucosal plexus, which is present between the submucosa and the muscular layers, right? So you have two plexuses, which I told when I showed you the structure. I told you about these two plexuses. One was the myenteric plexus, which is located between the longitudinal and the circular smooth muscle layer, that is your muscularis external, and second was the submucosal plexus, which is present outer to the submucosa, between the submucosa and the muscle layer, right? So here you can see that the fibers are mainly ending on the neurons, they are synapsing with the enteric nervous system. Then of course you also have sensory fibers, afferent neurons भी होंगे जब आप innervation की बात करोगे. Then you also talk about the afferent neurons, right? So afferent neurons would be arising from the epithelial structure also. And how would they be stimulated? They would be stimulated by the contents of the lumen. It can cause distension of the lumen. If there is contents within the GIT, it will cause distension. It can cause tactile stimulation. It can cause chemical irritation. So all these things can also lead to excitation, which would be, or it could lead to signals which would be carried by the afferent or the sensory neurons. Right? So what is the difference between the two plexuses of the enteric nervous system? ये बहुत important question है. So this question almost comes every year. If it is not coming as a short note or a, it will definitely come as your MCQ. Okay. So Enteric or Orbach's plexus is the outer plexus. Submucosal is the inner plexus. This I have already told you the location. Stimulation, it causes the tone of increase in the tone of the gut wall, increased rate of contraction, increases the rate of peristaltic waves, increases the rate of rhythmical contractions within the GIT. Submucosal, because it's present in the submucosa where your secretory glands are located, so it is mainly sensory in function. It controls the secretory activity and blood flow to the gut. So that was your efferent component. Now we come on to the afferent sensory fibers which are present. These I have already told you that these are stimulated by irritation of gut mucosa, distension of gut, presence of specific chemicals within the gut. They convey signals to both the nerve plexus. And what is their role? They can elicit either local reflexes, मतलब वो within the GIT से गया, enteric nervous system पे synapse किया और वहां से ही उसका efferent component निकला जो effector organ पे गया. तो वो तो एक local reflex हो गया. ठीक है. दूसरा क्या होगा कि वो GIT से गया, कहाँ गया? उसने enteric nervous system में भी इनपुट भेजा और उसने हायर सेंटर में भी इनपुट भेजा. हायर सेंटर यहाँ मतलब हुआ एक्सट्रिंसिक इनोवेशन पे गया. ठीक है. कि वो पैरासिम्पैथेटिक न्यूक्लियस पे चला गया या सिंपैथेटिक एक्टिविटी को ऑल्टर करा. राइट. इन एडिशन टू दैट, ऑल ऑफ अस आर फेमिलियर विद कि जब भूख लगती है, मतलब यू नो you could be in a certain state of emotion where your gut activity might increase or it might decrease. बहुत ज़्यादा anxiety हो जाती है, तो you can also have diarrhea. Some people can have constipation. So you know there is a connection between our gut and the brain, which is mainly known as your gut-brain axis. So brain is sending signals to our gut and gut is sending signals to the brain. So both of them are modifying each other's activity depending upon what is the state, what is our mental condition. When you are very hungry, then also you cannot concentrate because there are signals which are moving from your gut and affecting our you could say the emotional state or the state of our brain, right? So sight, smell, taste of food. You want to, of course, you must have already had your lunch at the moment. You must have reached home and had your lunch. So you might not be that hungry at the moment, right? So, but अगर एकदम घर पहुंचो, सुबह से कुछ नहीं खाया, right? And you smell something good food. You smell a food which is towards your liking. So your mouth starts salivating. Salivary secretion increases. So this is again an example of how the sight, smell, or taste of food can affect the activity of the gut. Or you have ingested something, so there is the role of your chemoreceptors, the osmoreceptors, the mechanoreceptors, which will have a role on the nerve plexus, on the enteric nervous system, and it will affect the activity of the smooth muscle or the gland. So this is your short reflexes and this is the long reflexes. This is just a summary of the innervation of GIT, basically how it is acting, how there are connections. between the extrinsic nervous system and the intrinsic nervous system, how there are connections between the GI and the nerve plexus, how there are connection between GI and the central nervous system, and how the higher function, other emotions and the other sensory inputs affect our GI function. Last thing in the structure and function of GIT is your GI blood supply. Again, splanchnic circulation का आपका पूरा एक lecture हुआ होगा. So, splanchnic circulation here, you can see that this is the abdominal aorta which is coming with the various organs of the GIT supplied by branches in the form of celiac artery, superior mesenteric and inferior mesenteric artery. Now, what is peculiar about the GI blood supply is that it does not directly reach the heart, but by these organs it flows into the portal vein. Portal vein flows into the liver, and in the liver the sinusoids are going to remove the toxic metabolites, the toxic substances which are present there, and then by means of hepatic veins it will flow into the inferior vena cava, right? And of course the liver is supplied by the hepatic artery. So the role of gut activity and metabolic factor on GI blood flow: mucosal blood flow is greater as compared to muscle. Capillary filtration coefficient is 10 times more than that of muscles. Willis has countercurrent system, जैसे आपने renal system में पढ़ा है वैसे small intestinal villi में I showed you the diagram which has a presence of the arterial and the venous vessels within it. So there is a shunt in the form of a countercurrent system. Blood flow in each area of the GIT and each layer of the gut is related to the level of its activity. ठीक है, अगर activity ज़्यादा होगी तो वहाँ पे blood flow ज़्यादा required है, और अगर वहाँ पे activity कम है तो blood flow कम required है. Increase in motor activity, increase in blood flow to the muscle layer. Increase in absorption and secretion will increase the blood flow to some mucosa and villi. So depending upon activity कहाँ है ज़्यादा, the blood flow to that particular layer will increase. This I have again, parasympathetic stimulation increases the blood flow, which is secondary to increase in the glandular activity. Sympathetic stimulation decreases the blood flow, and in case the blood is required, the circulation required, the vital organs require more blood, so it would be bypassed from, or it would be taken away from the splanchnic circulation and provided to the vital organs of the body, that is your brain, the heart, the kidney would require. But there can also be an autoregulatory escape which occurs अगर metabolic vasodilators बहुत ज़्यादा इकट्ठे हो गए within the GIT, then it can lead to an escape in the decrease in the blood flow. So these are the various hormones which increase the blood flow. These I will be dealing with when I am dealing with individual hormones. Increased vagal activity, I have already told you, increases the causes increase in the blood flow because of the increase in the glandular activity. Bradykinin is a vasodilator, so it again leads to increase in GI blood flow. If there is a deficiency of oxygen, which leads to release of adenosine, it again is a vasodilator. Right, so that completes your first competency, that was the structure and function of the gastrointestinal system. So in that, the gastrointestinal system, what does it include? It includes your tract, it includes the accessory glandular structures. In the structure, you should be familiar with the layered structure of the GIT, the variations which are there in the layered structure of the GIT, the importance of each layer of GIT, what is the blood supply of the GIT, what is the nerve supply of the GIT. In the nerve supply of the GIT, what is the function of the autonomic nervous system and what is the function of the enteric nervous system. In the functions of the GIT, you have the digestive and the non-digestive functions of the GIT. In the non-digestive, you have the protective function, and in the digestive, you have the digestion, the absorption, the secretion, the elimination, and the motility or the movement, right? So, I'll just start this next topic in 2 minutes. So I'm just giving you a 2-minute break. Don't log out. Any questions you can communicate through your CRs to me, okay? Okay, so I'm starting off with the next competency. Now, second competency in GI system is describe the composition, mechanism of secretion, function, and regulation of saliva, gastric, pancreatic, and intestinal juices. Now, each of this is a long topic, only the salivary secretion is a smaller one. So today I will be dealing with the first part of it, that is the salivary secretion. Okay, so I'll be dealing with the salivary glands and its secretion. So this again, there are various anatomical types of glands in the GIT. एनाटॉमी में आपने पढ़ा होगा, सिंगल सेल होते हैं. Then you have what is basically single cell glands. These are also the mucous glands, or simply called the goblet cells. These are located on the epithelium of the GIT, and their main function is to produce or to secrete mucus. Mucus forms a protective covering also, and it gives the it helps in lubrication of the contents also. Second type of glands which are present in the GIT are the tubular glands. These tubular glands, these are basically present in the stomach and the upper duodenum, and these are long, as the name suggests, long tube-like structures. They are primarily present in your stomach and duodenum, where they secrete the gastric acid. Third type of glands are, when I was telling you about the structure of the GIT, I told you that the epithelial lining in the small intestine, it is thrown into finger-like projections that are known as villi. In between them, you have presence of the crypts. These are the crypts or the pits of Lieberkühn, and these glands are present, these types of cells are present in the indentations in the epithelium or in the crypts, right? So again, you have various types of cells which are present in the crypt. And next is the complex glands. These are your associated GI glands, which includes the salivary, the pancreas, and the liver. There are certain principles before I go on to the salivary secretion. When I talk about the gastrointestinal secretions, so there are certain general principles of all forms of gastrointestinal secretions. So gastrointestinal secretions, these are secreted from the secretory glands, and they have two main functions. One is that because they contain the digestive enzymes, so they help in the digestion of the various food stuffs or various nutrients. Okay, so one is that they have the function of secretion of digestive enzymes. Second is that they provide mucus, which comes from the mucus gland, and mucus in turn has a number of properties and importance. The next slide would show that. Okay. Another general principle of gastrointestinal secretions is that the secretion in each segment of the tract is only to the tune of how much is needed there, right? ऐसा नहीं है कि say it requires a secretion of 1 milliliter per minute, so it will be secreted at the rate of 10 milliliters per minute. No, the quantity which is secreted in each segment will be exactly equal to, almost exactly equal to how much is required. Another general principle is that the constituents of secretion will vary in different portion, and even the type of food which you ingest is going to determine what the secretion is to be. So say suppose you have taken in a fatty meal, तो ऐसा नहीं है कि आपने fatty meal ingest करी तो प्रोटीन के एंजाइम्स ज्यादा सिक्रीट होंगे, क्योंकि फैटी मील इंजेस्ट करी है, सो यू नीड the enzymes which are required for digestion of fat. So that is those are the enzymes which are going to be secreted by our gastrointestinal system. So the first thing in the first secretion in the gastrointestinal secretion is mucus, which will be common to almost all areas of the GIT, right? So what is mucus? What is the, now you can get a question on what is the physioclinical significance of mucus secretion in the GIT. Okay, what is the physioclinical significance of secretion of mucus in the GIT, or it could be what is the physioclinical significance of mucus secretion in the body. तो फिर आपको ये ध्यान रखना है कि mucus कहाँ secrete होता है, because as you must be knowing that mucus is also secreted in your respiratory system, right? So you have to read the question very carefully and answer accordingly. So what is the significance? उसके लिए आपको ये पता होना चाहिए कि mucus after all होता क्या है, उसका composition क्या है, उसकी properties क्या है. So this is showing you basically a simple mucus secreting cell. So here you can see that this is a mucus cell and it has released or secreted mucus into the lumen and it forms a protective covering. What is mucus? It is a thick secretion comprising mainly of water, electrolytes and glycoproteins. It has various properties and each property is serving a particular function. So the first function of mucus is, or the first property of mucus is that it is adherent. Adherent means that it can stick to something. So once it is sticking to something, it can tightly adhere or stick to the food and other particles, and it can spread as a thin film or it can spread as a surface lining over the surface, right, over the surface epithelium. So it is adherent. First property is adherent. Second is it has sufficient body. What does it mean? कि उसमें quantity काफी उसकी उसका area exactly नहीं बोलेंगे. It helps in, again, this property helps in coating the wall of the gut so that the food particles will not be contacting directly the layers of the GIT. So it is again serving in the protective function. Third is that it has low resistance. So once it has low resistance, what will happen? The particles can slide over the epithelium. So it is helping, it is kind of a lubricant. It is helping in lubrication. Self-adherent, again, of course, that is the same as adherent and self-adherent. So they adhere to one another. And this is mainly, you could say, is more important when we talk about the feces. So it helps in sticking together of the fecal particles, right? So self-adherent mucus particles will help in sticking together and they play a role in the adherence of the fecal particles. It is resistant to digestion by the gastrointestinal enzyme. So again, it is offering a protection to the GIT, and it has buffering capacity because of its amphoteric properties. So depending upon which part of the GIT it is secreted, it can neutralize the acid as well as alkali to a certain extent. Now these GI secretions, they will be controlled. How the secretions of GI tract will occur? They would be under the control of a number of signals. It can occur when you ingest a meal, and it could be in between the meals. So either it is in the fed state or it is in the non-fed state, that is the interdigestive period. So depending upon whether it is the secretions occurring in response to a meal or in response to the period in between ingestion of the meal, when maybe you are starving or you are fasting. So the secretions which would be having a different type of characteristic as compared to when we have eaten a meal. So there can be various signals like chemical, osmotic, mechanical. There can be local signals, tactile, distension of the lumen, the irritation which can occur because of the contents of the GIT, which will lead to control the secretions from the GIT. Then there are specific substances which are released whose action on the secretory cell causes secretion. These are known as secretogogues. These are known as secretogogues. For example, you have GI hormones like gastrin, cholecystokinin, pancreazymin. So all these would be affecting the secretion of different types of GI secretions. Third could be control on the GI secretions, as I've already told you, is the nervous input, the parasympathetic and the sympathetic stimulation. And this table is giving you how much GI juices are secreted from which part of the GIT. So this is, it's required because it can be asked from you all in your Viva Voce, and of course it can form a part of your MCQ. The book में दिए हुए हैं, okay, all these are given in your books. So I've just picked it up from the books itself. Now coming on to the physiology of salivary secretion. So salivary secretion is of two major types. One is which contains the salivary enzyme, that is the salivary alpha amylase, also known as ptyalin, and this type of secretion is your serous secretion. Another is of course which contains mucus or mucin, and it is for lubricating and for protection of the surface. This is how much saliva is secreted, how much could be the maximum rate of salivary secretion. Salivary glands, again this is a revision of your anatomy. You have three major pairs of salivary glands. You have the parotid gland with its duct, you have the submandibular gland along with its duct, and you have the sublingual gland along with its duct. So the parotid duct, it is known as the parotid or the Stenson's duct. The submandibular duct is known as the Wharton's duct, and the sublingual is your ducts of Rivinus or the Rivinus ducts. You have 5 to 15 small ducts of Rivinus. ये याद कर लेना कहाँ से इनकी opening होती है, where they are opening, what they are secreting. So this is just a small table which shows that what are the major differentiating features between the three main types of salivary glands. What is their histologic type, what kind of secretion is occurring from this, how much of the total saliva, percentage of total saliva is being contributed by each type of gland. So this gives you a total of 95%. The remaining 5% is coming from the lingual and other minor glands which are present in the oral cavity. So main secretion of saliva is coming from the submandibular gland, which is both serous as well as mucus. Parotid secretes only serous kind of secretion, and sublingual is mucus secretion. Histology of salivary glands. When you talk about a secretory gland, this is a complex type of gland. It falls in the category of a complex type of gland. It has acinar cells and it has ductal cells. It has the acini. They contain two types of cells. One is the serous cells, which would be secreting the salivary alpha amylase also, and second type of cell is the mucus cells, which would be secreting mucin. Now all these cells, these are the epithelial lining of these cells. Then these would be communicating, or it could be interacting with one another by means of the interlobular ducts. These interlobular ducts will combine together to form interlobar duct. Interlobar duct would open into the main duct of that particular gland. Right? So you have the presence of serous cells, you have the presence of mucus cells, you have the presence of epithelium, then you have the presence of a central lumen. This is where the secretion would occur. First, the acini will secrete the primary secretion, which would be modified by the ductal cells, and then what will flow out would be the final secretion from that particular salivary gland. So innervation of salivary glands, again it is afferent as well as efferent innervation. Efferent comes from parasympathetic and sympathetic. Parotid is supplied by the glossopharyngeal nerve. The rest of the two glands are supplied by the facial nerve. Sympathetic innervation is from the thoracic first and second segment of the spinal cord. Afferent innervation is by means of the 7th cranial nerve and the 9th cranial nerve. Okay? So the chorda tympani branch of the 7th cranial nerve is afferent, and of course you have the glossopharyngeal nerve again, which has the afferent neurons within itself. So this is your innervation of salivary glands. This is showing you the salivary gland, this is the parotid gland, this is the submandibular, this is the sublingual, right? This is showing you the afferent side. Afferent side mein you can see there is the 7th cranial nerve and there is the 9th cranial nerve. Now these go to the two nuclei, the superior salivatory nucleus and the inferior salivatory nucleus. Inferior salivatory nucleus is for the 9th cranial nerve, which gives efferent fibers to the parotid gland. The superior salivatory nucleus gives rise to the facial nerve fibers, and here it goes to the submandibular and the sublingual glands. Composition of saliva, it contains mainly water, it contains solutes which can be inorganic and organic. So ye sare again you have to, because this is your core competency, so you need to remember these. If you can remember the composition in terms of how many milliequivalents is present, well and good, but otherwise itna to aapko aana hi chahiye, okay? Again, this is given in your books, so you can see it from the books also. Ionic composition of saliva depends upon the rate of secretion. This is showing you the ionic composition in the resting state. Okay, when you have not eaten anything or when there is no stimulation, then also there is a resting rate of secretion of saliva, wherein the concentration of sodium and chloride is much less as compared to the plasma, and of potassium and bicarbonate is much higher as compared to the plasma. Saliva is hypotonic, and loss of saliva from the body can lead to depletion of the potassium ion, because you can see that potassium is in a much higher concentration as compared to that of plasma. Functions of saliva, again important, you can get it as a short note, although this is a simple topic, but questions do come from GIT. So in your paper 1, GIT would be there in your paper 1, and in paper 1, like you know, you would be having 10 MCQs and you would be having a total of 100 marks paper. Each MCQ carries 2 marks, so GIT से आपके कम से कम 2 MCQs आएंगे, and in the remaining paper you would get questions worth at least you could say 10 to 20 के बीच में. So total of 15 to 20 marks would be devoted to GIT. Okay, तो अगर GIT से question आता है, so these are the few questions. So you can get a question on physiology of salivary secretion, or you can just get a short note on functions of saliva. Again, lubrication, binding, solubilizes dry food, so serves as a solvent for molecules which stimulates the taste bud. It is essential for maintenance of oral hygiene. It protects the tooth enamel. It washes the mouth. It destroys the bacteria, and there is presence of lysosome, thiocyanate, antibodies, all of which help in destroying the bacteria. Mechanism of secretion of saliva. It occurs in 2 stages. As I told you, it is a primary secretion which will be secreted from the acini, and then there would be modification which is going to occur within the gut, which is known as reconditioning of the secretion in the duct. So in primary secretion again depending upon you are talking about the parotid gland, submandibular gland or the sublingual gland. So here this is basically because showing you secretion of both ptyalin and mucus. So you could say it's the mixed secretion, which is the submandibular secretion. Once it is secreted, the composition of these ions is same as plasma. So in the primary secretion, the presence of sodium, potassium, chloride, bicarbonate ions in the primary secretion whose concentration is almost similar to that of plasma. जब ये डक्ट में फ्लो करेगा तो यहाँ पे एक्टिव सिक्रीशन होगा और एक्टिव एब्जॉर्प्शन होगा, पैसिव एब्जॉर्प्शन और सिक्रीशन भी होगा जो इसको मॉडिफाई करेगा. सो इट इज नोन एज रिकंडीशनिंग ऑफ द सिक्रीशन इन डक्ट. कैसे होगा रिकंडीशनिंग? सबसे पहले I'll go back to that slide later on. Okay, so what is the mechanism of secretion of saliva? So here you can see that this is showing you the first step in the secretion, how the primary secretion occurs. So these are the acinar cells. In the acinar cells, what will happen is that there would be secretion of the sodium ions, there would be secretion of chloride ions, and because the sodium ion and the chloride ion are being secreted, there would be the you could say that the reabsorption of the potassium ion would be there, right? So primary secretion, what will happen is that sodium is actively secreted from the acinar cells into the lumen of the acini, right? Once it is secreted, इसका जो movement है is much more पॉजिटिव आयन है, तो इससे क्या होगा कि यहाँ पर पॉजिटिविटी क्रिएट हो जाएगी. जब पॉजिटिविटी क्रिएट हो जाएगी तो क्लोराइड उसके साथ मूव करेगा. जब सोडियम और क्लोराइड मूव करेगा तो क्या होगा कि यहाँ पे कॉन्सेंट्रेशन सोल्यूट का बढ़ जाएगा. सो व्हाट विल हैपन, वाटर का ऑस्मोसिस विल अकर. सो द सिक्रीशन ऑफ अ प्राइमरी isotonic fluid which is rich in sodium chloride occurs in the acini, from the acinar cell into the lumen. Now once this occurs, it flows into the duct. In the duct, what happens is that there is an active reabsorption of the sodium ions, and along with the active reabsorption of the sodium ions, the potassium ions are being actively secreted in exchange for the sodium ion. So सोडियम का concentration कम हो गया, पोटेशियम का concentration बढ़ गया. Now what will happen कि सोडियम का जो absorption है is much more as compared to the secretion of the potassium ions. So again the chloride will move along with it. Bicarbonate का by and large they would be secreted by the ductal epithelium into the ductal lumen. Okay. So all this movement is occurring by means of specialized channels, or it is occurring by means of specialized pumps which are present in the acini and in the duct of the salivary glands. So two major processes occur within the duct. Sodium is actively reabsorbed, potassium is actively secreted, because of which there is passive absorption of chloride ions. Second is bicarbonate ion secretion occurs by ductal epithelium. This is passive exchange for chloride ions, and there is also an active secretory process. Final secretion of saliva is hypotonic because the ductal epithelium is relatively impermeable to water. I hope it is clear, right? So you have the primary secretion. Primary secretion involves secretion of sodium and chloride. This sodium and chloride is accompanied with osmosis of water, as a result of which there is isotonic fluid within the lumen. Once it reaches the cells in the duct, the sodium is reabsorbed actively, potassium is secreted, chloride is reabsorbed, and bicarbonate is secreted. Water is reabsorbed to a lesser extent, so it becomes hypotonic in nature. अगर इसका रेट salivary secretion का increase हो जाएगा तो ductal reconditioning will not be able to occur, or it will occur to a lesser extent. So what will happen? There would be loss of more and more sodium and chloride in the saliva, and the potassium and bicarbonate concentration will decrease in the saliva. So as the flow rate, now this you can see, this is showing you the plasma concentration of sodium, chloride, bicarbonate and potassium. So this is the rate, the flow rate of secretion of saliva. As the rate of secretion increases, you can see that the concentration of sodium increases in the saliva, the concentration of chloride increases in the saliva. Bicarbonate also increases to a slight extent, but by and large the potassium decreases in the saliva. So ions approach their plasma values because less time is available for modification, and bicarbonate remains elevated because salivary agonist stimulates specifically its secretion, right? So जैसे जैसे secretory rate increase होगा saliva का, उसका composition differ करेगा. क्या differ करेगा? क्योंकि primary secretion का concentration plasma जैसा है, तो इसके जो concentration of these four ions है would by and large start approaching the plasma levels of sodium, potassium, chloride and bicarbonate. Then we come on to the regulation of salivary glands or regulation of salivary secretion. So this is just showing you the parasympathetic innervation of the salivary gland. Now this I've already shown you. I showed you with the help of a different diagram. So this is just a repetition of it. Here you have the submandibular gland and you have the sublingual gland, which is supplied by the facial nerve, the fibers coming from the superior salivatory nucleus. Then you have the parotid gland from inferior salivatory nucleus. It is through glossopharyngeal nerve. The afferents are via the glossopharyngeal and by the facial nerve. They go to the nucleus of tractus solitarius. This is showing you the nucleus of tractus solitarius, which affects the activity of these glands. So the sight, the smell, the taste, the sound of food, higher centers will affect the salivatory nuclei in the medulla. It will also be affected by the sleep, the fatigue or fear can decrease its activity. The parasympathetics will cause increase in the secretion of saliva, both increase in the acinar as well as the ductal secretions, and there would be vasodilatation which will also occur as a result of parasympathetic stimulation. Again, just an overview of how these factors are acting. तो ये पूरी एक flow chart है. Salivatory nucleus in the medulla. It's just what was shown in the previous slide. उसका ही है. Sympathetic stimulation directly increases salivation. However, it can decrease acetylcholine stimulated salivation because it limits the blood flow. So here you can see that both the sympathetic and parasympathetic can increase. Sympathetic लेकिन क्योंकि parasympathetic को depress कर सकता है, so it can lead to decrease in salivation also. So the effect of sympathetic stimulation can be both stimulatory as well as inhibitory. Factors I've already enumerated. You have the taste stimuli from tongue and other areas of the mouth. Sour taste will elicit the copious secretion or high secretion of saliva. Smooth object will cause presence of smooth objects in the mouth will cause increase in salivation. Rough objects will cause lesser salivation as compared to smooth objects. Higher centers can increase it, or there can be reflexes which are originating from stomach and small intestine. So when I'm talking about the gastric juice secretion and intestinal juice secretion, I will also talk about the cephalic phase of gastric juice secretion. So basically when how the cephalic in that phase, there would be increase in the salivatory secretion. Blood supply. This again is a revision of what I told you. Blood supply to the glands ensures a supply of nutrients from the blood. Increased blood supply would be by parasympathetic stimulation. Salivation itself directly dilates the blood vessels. Then the kallikrein secreted by activated salivary cells causes the formation of bradykinin, which is a vasodilator, and sympathetic stimulation can lead to slight increase in salivation, which is rich in mucus. If there is decreased in saliva or deficient salivation, that condition is called as xerostomia. So xerostomia is decreased salivation, which can lead to ulceration and infection of mucosa, and it can also increase the incidence of dental caries. Dry mouth can be because of related to drugs. It could be because of some vascular disorder. It could be because of the radiation treatment or autoimmune disease. Another clinical condition which is here as far as which can also lead to the defects in salivation is cystic fibrosis. So cystic fibrosis, transmembrane conductance regulator. What happens here is that salivary calcium, sodium and proteins are elevated. So the chloride secretion is essential for secretion. Who lack the chloride transporter, there would be the ionic composition of the saliva is going to change. Another condition which can affect the composition of saliva is aldosteronism, primary aldosteronism. This aldosterone hormone which is secreted from the adrenal. This can again cause the excess of aldosterone. What happens is salivary sodium chloride, it can bring the salivary sodium chloride close to zero, because aldosterone is the hormone which leads to the reabsorption of the sodium. So what will happen is sodium would be reabsorbed, potassium would be secreted, and so there could be depletion of potassium in the body because of primary hyperaldosteronism, right? So that completes the physiology of salivary secretion. So in the salivary secretion, you should be knowing about the salivary glands, their histology, their structure, then their nerve supply, what is the composition of saliva, what is the mechanism of secretion of saliva, how the secretion of saliva is regulated by the nervous mechanism and by other stimuli. And you should be familiar with the applied aspect of it, in which primarily you should be knowing about the xerostomia, and you should be familiar with what will happen in to salivary secretion, or what will happen. Aldosterone will mainly come in your endocrines. Okay, so as far as your salivary secretion is concerned, you should be familiar with xerostomia and cystic fibrosis and aldosteronism. So I think I'll stop here. So any queries you can communicate through your CRs, right? And there is one more thing. I think 20th of October is a gazetted holiday, and I had a lecture on 20th of October. So I plan to take that lecture. Timings we can coordinate. Okay, you can communicate your convenience through your CRs. So I will not be missing my 20th October lecture. Otherwise, बहुत gap आ जाएगा, and we don't have much time now for your send ups also. Okay, so thank you all.